The overall goal of this procedure is to establish an objective method of quantifying damage to cotton plants caused by 2 spotted spider mites using an active multispectral optical sensor.
This method can help answer key questions in agricultural entomology, such as which insecticides are best able to control spider mites in cotton. The main advantage of this technique is that it provides researchers an objective metric for evaluating spider mite damage on cotton while saving time and valuable human resources.
We first had this idea for this method for evaluating different insecticides on cotton for efficacy against 2 spotted spider mites.
To begin the protocol, grow non-genetically modified or non-BT cotton plants to 4 to 5 true leaf stage in plastic trays in a greenhouse. Next, transfer spider mite colonies from the pinto beans onto the young 4 to 5 true leaf stage cotton plants. Transfer 3 masses of spider mites for lightly infested plants. Place a pan under the pinto bean leaf tip containing 2 spotted spider mite, or TSSM masses. Cut the pinto bean leaf tips with scissors, allowing TSSM masses to fall into the pan. Turn the pan upside down over the cotton plants and tap the TSSM masses onto the plants. Then randomly spread the 3 masses of the TSSMs onto the cotton plants. For medially infested plants, transfer 20 masses by placing a pan under the pinto bean leaf tip, containing the TSSM masses. Cut the pinto bean leaf tips with scissors and allow them to fall into the pan. Turn the pan upside down over the cotton plants and tap out TSSM masses.